the case at Delavan Grider. So, well, let me take this opportunity to get this show on the road and welcome you to the Mini Medical School. We have a good turnout this evening. In addition to our regular Mini Medical students who have enrolled in the Mini Medical School, we're also being graced with the presence of our Asian alumni who are joining us from China, India, and Indonesia, and it is the wee hours of the morning over there. So kudos to you for setting your alarm for Mini Medical School. We also may be joined by some students from Buffalo Prep, and this would be their first time joining us in Mini Med. So welcome to all of you who are in either middle school or high school. We are really thrilled that you're here to join us for this second in a series that are addressing some issues related to COVID. It's called A Second Dose of COVID, and as you just heard, one of our speakers has just gotten her second dose, Dr. Surtees. So I hope many of you, if not all of you, will be getting your first and second dose very soon if you haven't already. So I am Dr. Roseanne Berger. I am the Mini Dean of the Mini Medical School. I'm also the Senior Associate Dean for Graduate Medical Education and a member of the Department of Family Medicine, as is one of our speakers this evening. I am joined tonight by a number of people who are experts in their field, but before I introduce them, I wanted to give a special thank you to the Mini Med Advisory Board that was organized this year to help design the curriculum for Mini Medical School. We do follow the tradition of medical school in the sense that we combine some basic science with the application of that basic science to the clinical arena. And many of you may have heard the expression in terms of talking about research from bench to bedside. In Mini Medical School, we have changed that phrase somewhat, and we call it from lab to living room. So you'll see tonight how some of the scientific work that's done at UB is translated into things that can impact us on a daily basis. So now let me introduce our scientists and clinicians. First, we have Dr. Jen Surtees. Dr. Surtees is an associate professor in the Department of Biochemistry, and she is also the co-director of the UB Genome, Environment, and Microbiome Community of Excellence that we call GEM. She is here because of her expertise in genetics, and in particular, her work on the variants that are related to COVID-19. We also have Dr. John Selleck. He's a professor in the Department of Medicine in the Division of Infectious Disease. And relevant to tonight, he is the epidemiologist at the VA and Kaleida Health, and he is on an advisory committee that includes the Dean of the School of Medicine, and he's a constant resource when we have questions about the impact of COVID on our trainees. And lastly, we have Dr. Raul Vasquez. He also is a family physician and the Chief Executive Officer and founder of the Greater Buffalo United Accountable Healthcare Network. He's been in the news quite a bit because of the efforts that he has led in getting vaccine out to the community, as well as reaching out to the community for healthcare, which really is the mission of his organization. So we will be starting shortly with Dr. Surtees to start with our basic science. But I just wanted to give you a little preview of what will be coming in the months ahead. In April, we will have a presentation on promoting healthy aging through high intensity interval training. Many of us think of that for athletes, but it is good for those of us who are getting older. And Dr. Bruce Trone, a geriatrician, will be heading up that team of speakers. In May, we will be discussing the role of nutrition and good health outcomes. And then in June, we'll be looking at some really interesting discoveries that have been made by the lab that Dr. Tony Campanari leads, and it is involved with joint replacement surgery. And last but not least, in terms of thank yous, we could not do this without the contributions of our UBMD faculty practices, which are the sponsor of MiniMed for this season. So thank you all. And with that, I'm going to turn the podium or microphone or Zoom screen over to Dr. Surtees.
Thank you for that. And I will share my screen. So it's a real pleasure to be here this evening to talk about, you know, what's on all of our minds right now still, SARS-CoV-2, and some of the work that we've been doing on genomic surveillance of the virus in Erie County, and how that fits into a global perspective. And I just want to make sure that I tell you about the team of amazing people who've been working on this over the past year. So this is has been a collaboration with the Erie County Public Health Department of Health and the Erie County Public Health Lab. So Gail Burstein agreed to collaborate with us, and Carlene Pope is the person who gets us all of the samples that I'll be talking about tonight. And then Dawn Yurjo is the Associate Director of the UB Genomics and Bioinformatics Core, who has been instrumental in getting the sequencing up and running. Jonathan Bard has been instrumental in getting the bioinformatic analysis up and running. And Natalie Lamb has come in and is doing some more innovative bioinformatics as well, which is really exciting. And this is all done at the Center of Excellence in Bioinformatic and Life Sciences. And Norma Nowak is the executive director there. And she's also my co-director for GEM. We got funding to start this from a pilot project from SUNY Research Foundation, and subsequent funding to do all of this sequencing from GEM. So I just wanted to start by reminding you of the SARS-CoV-2 life cycle. So as you know, this virus is circulating around the world and in our community. It is a respiratory virus. And it is an RNA, genome, which is an important component. So we have these nice little virus particles, and they're coated in the spike protein, which we've all heard so much about over the last little while. And these spike proteins are able to recognize these receptors on the surface of our cells. And through this interaction, the virus enters into the cell as this viroid, and then breaks apart and releases the viral genome, which is this RNA sequence that is translated or converted into protein to allow for replication of the genome, that is copying, making many, many, many copies of the RNA genome, and also making all of the proteins that the virus needs to make new virus. So a virus particle gets into your cell and basically co-opts all of your cellular machinery to make more copies of itself. And then it releases itself back out into the world to infect the next person. One of the things I want to point out is during this replication process, this copying process of the genome, there is always, with every round of replication, there is a small but real probability that a mistake will be made in the copying process. And this is what leads to mutations. So the more infection you have, the more the virus copies itself, the more it's going to make these small mistakes leading to mutations. And that's really what we're talking about when we're thinking about variants in the SARS-CoV-2 variants. The other thing I want to point out is that all of this happens outside of the nucleus. So the virus doesn't go anywhere near your genome, your DNA. It's all outside of the nucleus, and then the virus gets released out into the wild. So what we've been doing in for the last year or so is doing whole genome sequencing of SARS-CoV-2. And so that is taking the RNA genome and decoding it so that we can understand how the virus is working. So how do we do this? So as I said, the RNA is, the genome is actually RNA, which is different from our double helix. And we actually don't have the technology to sequence RNA the same way we do sequencing DNA. However, biology has given us this amazing, these amazing natural reagents, which allow us to actually do reverse transcription, which converts the RNA genome into DNA. And so now we have a double helix, DNA double helix that we can sequence. So we amplify that by PCR, and then we do library preparation for next generation sequencing. So this is really high throughput and deep sequence. We can get a lot of information from this type of sequencing. And then we have to do sequence analysis. And so we go through this process from RNA sample from a COVID positive patient through to a string of letters, which is the genome that makes up SARS-CoV-2. And so if you think about an analogy for the genome, if you think about it as being a book that is the instructions for, in this case, the virus. It encodes all of the information that the virus needs to make more virus. And so when we sequence the genome, we're basically copying down all of that information, all of the letters that make up that genome, that make up that book. And when we have a mutation, that's going to change the letters. It's going to change the meaning in the book. And sometimes that meaning that change will be benign. We'll still be able to understand the plot. And in other cases, that change is going to be a bigger change and may change the plot or the meaning of the book. And that's kind of what we're looking for. So we get these sequences. And so far we've actually sequenced close to 1,000 genomes from Erie County of COVID positive patients. And when we get that sequence, we upload that sequence into a global database called GISAID. And I want to say that GISAID has actually been an incredible resource globally. Everybody who's doing SARS-CoV-2 genome sequencing is uploading their sequences to this openly available resource. There are nearly 1 million COVID sequences in GISAID right now. And all you need to do is register, get a user registration, and you can download those sequences and do what you want with them. One of the interesting things about GISAID is that in order to encourage data sharing, particularly from low and middle income countries, they actually require people who use the data from GISAID to make an effort to reach out to the people who did the sequencing in the first place, to collaborate with them, or to at the very least acknowledge them if they use that data in any of their publications. And so this is has been a way of encouraging open data sharing worldwide, and it's been extremely successful in this pandemic. So we upload all of these sequences. And so then what do we do with it? We just have these strings of letters. How do we make any sense of this? How do we use this to understand how the pandemic is progressing? So there's another website platform, called NextStrain. And we use this in conjunction with our data, as well as some custom pipelines that we make here. But what NextStrain does is allow us to build these phylogenetic trees. And this is really a way of displaying how closely related different samples, different patient samples are genetically. And I'm going to walk you through exactly how to interpret that in the next couple of slides. But when we have the NextStrain data and the GISAID data, that's all associated with time and place. And so in addition to getting a sense of how all of the sequences in Erie County, for instance, are related to one another, we can also work backwards and get a sense of where they came from and how they spread through the world using these sort of mutation fingerprints that we'll talk about next. So to read a phylogenetic tree, this is just a very simple example. What you look at is how close together your end products are. So here we have A, B, C, D, and E. And every time there's a branch point, that means there's been a new mutation that is allowed, that basically resulted in divergence from your starting point. And so here we start with this blue ball, which is a single mutation. And then there's a divergence here where this path, there's an accumulation of a new mutation. And here there's a different new mutation, and so on and so forth. When you have a line like this, it means that these two are identical. So A and B are identical, and they have the green mutation, the orange mutation, and the blue mutation. Whereas C, which is closely related to A and B, but not identical, because it diverged right here and it got this red mutation instead of the green mutation. But there's overlap. So they're genetically closely related. And then D and E are closely related to one another because they're coming off on this other branch. And they both have this teal mutation, but only E has accumulated this sort of darker red mutation. So they're closely related to one another, and A, B, and C are closely related to one another, but there is more genetic distance. If you think about family relationships, they're more distantly related. This group, A, B, and C, are more distantly related than from D and E. And so we can put all of our sequences onto this kind of tree and get an idea of how closely related things are. And so if you just look on the left side of this is some of our early samples, and the Buffalo or Erie County samples are in red, and they're overlaid on the global phylogenetic tree. And by looking at these relationships, we learned that so these are samples that took us through June. On the left are the very first samples, and then this is about 100 or so samples. But what we find is we have some samples that originated in Asia, but the vast majority of cases of COVID in Erie County originated from, or were a result of viruses that originated or came through Europe, which is similar to what happened in New York City, but different from what happened on the West Coast where the majority of their cases initially had an Asian origin. This is another way of kind of looking at, this is all Erie County samples. And so this is a phylogenetic tree, but it ignores the rest of the world and just looks at relationships within Erie County. And so for example, here we have a whole bunch of samples with identical genomic sequences. All of these sequences are de-identified for us, but the Erie County Public Health Lab knows who these people are, and they could find out if all of these people are from the same school or the same workplace, and could make decisions about public health as a result of making some of those conclusions. This is sort of more data. So over time, this is when we had a couple hundred or maybe 300 samples. And what we did here was just overlay on New York State. So the faint background is the phylogenetic tree for all of New York State. And then Buffalo are these filled circles that you see. And you can see that there's, and we're going through time. So the earliest samples are closer to the center of this bullseye, and the more recent samples are further out. And you can see that the Buffalo or Erie County samples are pretty similar in terms of the relationships that they have compared to the rest of New York State, with the exception of this part of the tree where we really are underrepresented here. So initially, when we were looking at these samples, we were really trying to understand how the virus got here, what route it took, and then also how it was spreading within the community. But early on in the pandemic, it became clear that there were going to be mutations that would affect the prevalence of the virus and the transmissibility of the virus. And the first major mutation that became dominant globally is this spike protein mutation, which changed this amino acid from a D to a G, aspartate to a glycine. And This was a mutation that arose in Europe way back in February of 2020. And this plot just shows you the proportion globally of the virus that had the original D at this position of 614 in orange, and the G mutation. And you can see it originated early in the pandemic and quickly took over. This G version quickly took over wherever it happened, wherever it was observed. And this was true through Europe and through the United States and throughout the rest of the world. And here in Erie County, we saw the same thing. So if you now we're just looking at whether we have a D or a G at this particular position. And you can see early on in the spring, we had a handful of viruses that had the original D, including one up here, but more of them had that G change, which was becoming prevalent worldwide. And if you look at our samples now, and this is taking us through to the end of January of 2021, we have no samples that have that D at this position. Everything is this 614G now. And this is, as I said, very similar to what's been happening worldwide. This change actually increased the transmissibility of this virus, and that's probably why it took over. If it was more easily transmissible, it was going to account for more infections. And then, of course, in the fall, early fall, we started to see these other variants of concern, which had increased transmissibility and in some cases, increased pathogenicity. And so these are just outlined here. The first was the so-called UK or United Kingdom variant. Although we're trying to use this nomenclature that is non-stigmatizing. So the B117 variant. This has several mutations in the spike protein, but I want to point out that all of these variants have mutations outside of the spike protein as well. In the genes that encode the machinery that replicates the genome and processes the proteins to allow viral formation and replication. The B1351 was originally identified in South Africa and also has both spike and non-spike mutations. And then P1, which was originally identified in Brazil, which has a lot of mutations throughout the genome. These are all considered variants of concern by both the CDC and the WHO. And these are, we call them variants of concern, but they're really a lineage with a constellation of mutations that when combined leads to increased transmissibility and increased disease in some cases. I want to make really clear, though, that with all of these variants of concern that I just mentioned, the vaccine has proven, the vaccines that are currently approved and circulating around the world have proven to be very effective at preventing serious disease and hospitalization, despite the fact that there are these changes within the spike protein on these viruses and these other changes that I mentioned.
Yes, this is the house phone. Yeah, you called it. So you're watching it like.
So, this brings us to January of this year, and starting to look at some of the variants of concern here in Erie County. So, unfortunately, we do have some of these variants of concern circulating here. Actually, just last week, we found 5 of the California variants, so-called California variants, which are these B1427 and B1429 variants, which are variants of concern according to the CDC, and the WHO considers them variants of interest. That's where we're watching them. But early studies indicate that they have about a 20% increase in transmission, and they reduce the neutralization by some of the monoclonal antibody treatments that have been approved in the United States. And so this has implications for healthcare choices. I'll also point out that The Wadsworth was doing some sequencing from Kaleida health samples and found the B117 variant, so the so-called UK variant, about a month ago. And actually just today they uploaded another B1427 variant from Erie County. So variants are here and they're circulating, and this is something that we need to really be aware of and take into consideration when we're thinking about our choices and what we do out and about or not out and about in the region. One and other variant is this P2 variant, which is a variant of interest, which is related to the P1, but we don't know very much about it at this point. So I'm going to stop here, and I just want to finish by saying that we have this infrastructure and partnership with Erie County Public Health Labs. Kaleida Health is starting to send us some samples as well this week. And we have this infrastructure for genomic surveillance of SARS-CoV-2. And now we're thinking ahead to the idea of genomic surveillance as a more common public health tool for other types of outbreaks and pathogens. And so I think this is a really powerful approach for dealing with outbreaks going forward.
Thank you. The work that you're doing is really helping to arm the clinicians as well as those of us who live in the community about the fact that we still have to be concerned about how this pandemic is evolving. You did touch on some of the questions that the audience has already submitted. I just wanted to clarify that we have a number of questions that were submitted by registrants in the conference in the Mini-Med School, and we will be getting to those after Dr. Selleck gives us a little bit of an introduction to how all of this science gets applied in the world of infectious disease, and in particular when considering how to utilize vaccines and how effective the vaccines are. You can put some questions in the chat. If we don't get to them during this hour session, we will prepare some responses that we will post on our Mini-Med page. But we do have quite a number that were submitted in advance. So Dr. Selleck?
Thank you, Dr. Berger, and thank you, Dr. Surtees. Tough act to follow. I just looked in my iPhone. It was about a year and 2 weeks ago, March 10th of 2020, that I met with those of you who are regulars at Mini Medical School. That was our last live in-person Mini Medical School, where I first gave you a little bit of hint of what was going on with the pandemic. And it was very shortly after that that the coronavirus hit the fan, and it's been a pretty incredible year since then. So when Dr. Berger sent me the first email and said, you know, we want to do Mini Medical School, and we're talking about a second dose of COVID. And that was when we were having all these goofy discussions about whether we should just give people one dose of vaccine and, you know, spread it around more. But she assured me that was not the intent of the region of the discussion. But again, I'll still take the opportunity to say, please get your vaccine. Please, please, please. You know, if you didn't believe it before, now hopefully you believe it more after what Dr. Surtees showed you. Now, the thing that's going to be interesting here, this is probably in 40 some years of teaching, the absolute shortest slide set I have ever made in my life. So what I wanted to show you that among the many questions that I get from people on a day-to-day basis, in fact, from one of the administrative people at Buffalo General this afternoon, should I get one vaccine or should I wait for another vaccine? So what I'm showing you here are the 3 vaccines that currently have emergency use authorizations in the US. And the 4th one should be coming along relatively soon. And I'm also showing you just the data from the trials that were done in the United States. So if you look at these over on the left-hand side, I've shown you what the vaccines are, and I put them in the order that they have been or will be approved. So the BioNTech, Pfizer, Moderna, Janssen, which is a subsidiary of Johnson & Johnson, and AstraZeneca. You can see that 3 out of the 4 are 2-dose vaccines. I wouldn't be totally surprised if the Janssen vaccine will eventually have a 2nd dose as well. And if you look at the total number of subjects in those trials, it's a little over 147,000, more than half of whom got vaccine. Some of the trials were 1-to-1, so 1 placebo, 1 vaccine. Some of them were 2 vaccine, 1 placebo. Their endpoints were all a little bit different in terms of how they defined clinical outcomes and clinical success. And the middle column is the one that everyone is extremely focused on, and that is the overall efficacy versus symptomatic infection. And you know that we were the pre-trial setup that was done with the FDA was that if a vaccine was 60% effective, we were going to be extraordinarily happy. And as you've seen many people like Dr. Fauci and Dr. John TV, the first 2 were just absolute home runs, the BioNTech, Pfizer, and the Moderna, the J&J and the AstraZeneca a little bit less. The Janssen J&J, they've also noted that they have 86% efficacy versus severe disease. But I really don't want you to get into that because, this is like, everybody's worried about SAT scores and MCAT scores and batting averages and all that kind of thing. What I want you to do is look over here on the right-hand side. And that is, the 2 places that you don't want to be if you get infected with SARS-CoV-2 is in the hospital or in the morgue. And all of these vaccines keep you out of the hospital and out of the morgue. So my response to everyone who asks, I can only get an X vaccine, should I do it? The answer is whichever one you can get put into your arm, please do it and do it as soon as you can. Now, there's a little bit of nuance here. As you've seen often on TV, the BioNTech, Pfizer, and Moderna's are the kind of bare messenger RNA, and they're put into a little lipid carrier. These have to be in the deep freeze. That's minus 70 Celsius. And that's all the discussion you've seen about these special freezers. They're not really special in a research lab, but most other places they're special, and they have to be handled appropriately and used expeditiously once you take them out of the minus 70. The Janssen AstraZeneca vaccines can be kept at refrigerator temperatures, which has a huge advantage for being able to deliver these, particularly to areas that are outside of urban areas where you may have ready availability of these minus 70 freezers. Excuse me, again, as I noted, the AstraZeneca vaccine is not yet authorized in the US. The reason that these other 2 vaccines don't require the deep freeze is they are what's called an adenovirus vector vaccine. So a particular adenoviruses infect humans and many other animals, and there are just hundreds and hundreds of strains. So these strains of adenovirus are turned into essentially a neutered carrier to bring the spike protein gene to your cell, where just like with the mRNA vaccines, your own cellular machinery will crank up and make the and make the spike protein, which your body then, your immune system then recognizes. The good thing about these vaccines, historically with vaccines in the US, it tends to be a lot of, 25-year-old males in vaccine trials because the feeling of the pharmaceutical companies, you don't have to worry about pregnancy. If you screen people adequately, everyone is healthy. But the decision was made that we were going to look at a whole population kind of setting. So you can look at the results of each of these vaccine trials, and you can see that it's spread out by age, it's spread out by geography, it's spread out by ethnicity, people with comorbidities, other medical problems like diabetes and heart disease were not excluded. So this gives us a lot of reassurance that that big Easter egg that I've given you there with all those 100% in it really applies to a broad population and not just to healthy young men. So the other question is always, how come we don't have data on pregnant women? And again, by and large, pharmaceutical companies have not been anxious to do studies on pregnant women for fear that if something goes wrong, the lawsuits will be absolutely crushing. But as you probably saw within maybe the last 2 weeks, that when you have 147,000 people in trials, there were some women who became pregnant during the trial, even though they were not pregnant at the beginning of the trial. And the result of the analysis of those women was that they had no problems with their pregnancies and that the babies were born perfectly healthy. And in fact, good evidence that the babies were born with immunoglobulin that moved across the placenta to protect them from infection. You've also seen in the last few weeks that most of these companies have now started trials. Generally, what they do is they start out with the adult population, and then once they see that everything is good in terms of side effects and generating antibody, then they start walking it back. So, Pfizer originally did 16, and Moderna did 18 years old. And then they went down to 12 years old. And now you're going to be seeing these 6 months to 12-year studies being done as well. A number of those are already accruing patients. The hope is that perhaps by the start of the next school year, that we will have vaccine approved for the older age group of kids, and then maybe by a year from now, we'll have vaccine that we'll be able to give to all kids, which will be excellent in every respect. So just a few key points. People say, and unfortunately, calling the project Operation Warp Speed, kind of made it, took us into Star Trek and Star Wars. Safety was not bypassed, absolutely not bypassed. All of the information was available online to anyone who wanted to read it before the FDA review committees, the VRBPAC, you've heard many people say that on TV, the Vaccine and Related Biologicals Advisory Committee, and the CDC's Advisory Committee on Immunization Practices. You can get your hands on all of the documents, and if you want, you can do your own statistics on them, et cetera. The RNA technology and the adenovirus vector technology has been available for several years. We just haven't used it in very large quantity for most of our infections. The adenovirus vectored vaccines were shown to be very, very effective for Ebola, and the ones that are being used are adenovirus vectors vaccines. Amazingly, no serious side effects have been reported in any of those trials, those 147,000 people. One of the things that you have to keep in mind, you saw that the UK said, well, we're going to put a halt on this for a little bit because we had 3 people with blood clots. When you use a population of people that includes people with underlying medical diseases, people who are older, some of them are going to have strokes, some of them are going to have heart attacks, some of them are going to have blood clots, whether or not they get placebo or a vaccine. So each time those show up, we look at the proportion of those occurring in the placebo group versus the vaccine group. And it is absolutely remarkable that we've had no serious side effects from these trials. The vast majority of vaccine side effects happen in about the first 6 to 8 weeks. So we're well beyond that point. Now, you have seen that there are rare serious allergic reactions to the mRNA vaccines, maybe related to the polyethylene glycol that's in this little lipid sphere that it's put in. I checked today at my 2 shops and at the VA with, we're now up to about 25,000 vaccines given. We have not had a real anaphylactic reaction. And at Kaleida Health, also with tens of thousands of vaccine doses given, they've only had 1 anaphylactic reaction. So don't let this scare you away. And again, as Dr. Surtees mentioned, the messenger RNA does not get into our genome. It's I usually say when people are asking about this, I noticed that Dr. Surtees did not grow horns or scales since she got her second vaccine dose today. And all of the people that I know, and including myself and my family, none of us had any of those things happen. So very, very safe. The duration of protection, don't be stuck on the 90 days. That is just based on original information. Undoubtedly, the protection is going to last longer. We already have good data from clinical infection that it probably goes to the 6 or 9 months range, and we will see that number continue to get stretched out. So the question that everybody asks is, how long is immunity going to last? We cannot answer that. We've only had these vaccines for a few months, but I think the general expectation is that we're probably going to have adequate antibodies for maybe a year or 2, and we're hoping that the T cell immunity, which is a part of the immune system that kicks in when particularly viruses get to our cells, will last even longer. And we now have, for those of you who keep up, yesterday CDC put out a special MMWR version showing that the 2 mRNA vaccines greatly decrease carriage and transmission, and Janssen and J&J have already shown that for that adenovirus vector vaccine. So I think we're getting multiple wins out of this. So again, please get vaccinated. Don't worry about variants yet. The variants that we've seen so far are not completely able to escape vaccination. So don't think that this is going to be futile. So please get yourselves vaccinated as soon as you can. I know it's a little bit of a challenge, but get yourself online and start hitting multiple websites, and hopefully you'll be lucky in a fairly short time period.
Dr. Selleck, you really anticipated a lot of the questions that have already been in the chat or have been submitted. But let's get to some of those questions. And just to reinforce your message, there are a number of questions about booster shots, which are related to the issue you were just talking about, which is the length of immunity. Do you have any prediction about the timing for booster shots? And also, would you anticipate that a booster vaccine will be a reformulated vaccine based on what Dr. Surtees was talking about, the emergence of variants?
Want me to go first? Whoever, I don't want to hog.
Jump in, whoever.
Dr. Estes, from my side, I just think now, because it's a platform, for how these vaccines were created, it's easy to piggyback on it. So if we do have to have a booster, it'll be a lot easier to create. You won't have to start from the beginning. I think that's what we tell people. But for now, you know, the vaccines that we have are doing a great job. I mean, we're seeing, you know, even the CDC changing platforms that after your second shot, 2 weeks after, you know, you don't have to, you know, isolate if you're exposed to COVID. If you're around people eating, I mean, you're going back to a little bit of a normal. So that's why I kind of encourage people that the vaccines are really important. Don't worry about the boosters right now. Let's worry about, you know, getting through this thing. And I think the more the data is being looked at, the more of these immunoglobulins, IgG, will know how long they're going to last. But that's just going to take time. And I think the studies are, as you know, it'll be 6 months, then it'll be 9 months, then it'll be a year. And my prediction is 5 years, you know, almost like we did with the measles, mumps, and rubella, the measles shot. That, you know, it's a little different formulated, but it does do a lot of things. And in addition, it's going to make us less infectious. So we're not just, you know, we're not going to get it, but we're also not going to give it to anybody else. And those studies are kind of, you know, eventually coming out.
Yeah, I think that, you know, the technologies that we're using here, the mRNA technology in particular, this is, you know, I hate lingo, and, you know, everybody overuses the lingo game changer. This is a game changer. You know, this really, you know, you can't turn this on a dime. But this is not, you know, not like most of our other vaccines. So in terms of how long do we think this is going to last, this is a virus that attacks you on your respiratory mucosa along the lines of influenza or respiratory syncytial virus or others. And for the most part, those vaccines do not give you the lifelong lockdown that you get with vaccines like measles and rubella, which spend a very long part of their infectious cycle in your blood, where antibodies are able to latch onto them and clear them out quickly. So most of our mucosal vaccines, as it were, do not give you that same kind of lifelong protection that we get with, you know, things like measles. But, you know, by and large, the people who do research with these are really hoping that we're going to get probably a couple years and maybe even longer. And a lot's going to depend on how much T-cell immunity these vaccines induce, because this, you know, again, this is going to be a little bit different than the vaccines that we've used before. So I think we're very hopeful that, you know, that this is going to last us. And then if we do get a major mutation that requires a change, it's relatively easy to turn the ship around and develop a new vaccine. But again, I wouldn't worry too much about the booster yet. You know, we're only a few months into vaccine. Please get the vaccine if you've not already gotten it.
And
So there have been Dr. Surtees, yes.
Jump in and really quickly say that they've actually already started looking at reformulated versions of the vaccine that are more towards the variants, and a couple of those are promising right now. It seems to be working, and not only that, the vaccines that they're testing against the variants also are equally effective against non-variants, and so there's a lot of overlap in terms of efficacy of the current vaccines that are out there and of the new ones that we're working on. And I will say globally, there is an effort to monitor and respond quickly using the influenza model, or starting to think about using the influenza model for predicting how we might need to change the SARS-CoV-2 vaccines.
Can you describe that model a little bit?
So every year there is genomic surveillance of influenza happening every year, and roughly 20,000 genomes of different flu positive patients are sequenced every year. And based on that sequencing, they choose which versions of those virus are going to be included in the seasonal flu vaccine that we get. And it used to be 3, now it's typically 4 different antigens that they use based on what's circulating. And they use what's circulating in the southern hemisphere to predict what's going to be in the northern hemisphere 6 months later. So
Thank you for that. Yes, I know Dr. Vasquez and I wait patiently every year to find out what the flu vaccine is going to be so we could get it in stock.
this year is going to be.
And what I wanted to say too is that, so it's good. So I'm a clinician. I want to get 75% of the population vaccinated. So in terms of the boosters and all that stuff, if we do that, then we're going to have the herd immunity. We're going to be, it's going to be good. But then we don't have to worry so much about these mutants and everything else going on. So, it's real important that people understand that, there's no virus in these things, especially the Moderna and the Pfizer. So, because in our communities, we don't have primary care doing this. We have everybody else but primary care. And I think when you don't have the connection, especially as we get into the Black and brown population, the hesitancy, and 40% of New York State is Black and brown. So unless we start to kind of get people educated to make these changes. So, and I know we talk about boosters and all that, that's all great, but we need to get the vaccine in people's
We need
arms.
the first round.
Yeah.
Yes.
Yeah. So there's been a number of questions also about individuals who have had COVID and have tested positive for antibodies, or maybe not even been tested for antibodies, but had the disease. And should they be vaccinated? And when should they be vaccinated? How soon after they have recuperated? And also related to that, if they have long COVID, in other words, if they have symptoms that are lasting longer than 12 weeks, does that alter the recommendation about vaccination for this group?
I think so. So I've given 4,000 vaccines out in the field. What we do is we wait till they get, if they had COVID, we wait for the time period for 2 weeks. And then a week or two after that, we've been vaccinating people. What I do tell people when we're vaccinating them for the first dose is that you may get symptoms. If you had COVID and didn't know you get COVID, you're going to have symptoms of COVID. You may have fevers, muscle aches. That'll last for about a day or two. Take Tylenol, Motrin during that time, and you pretty much do well. The second shot, as you know, then you start developing the memory cells and some of these symptoms. But with us, I mean, we're trying to get the shots in the arm. If you've had COVID, that doesn't mean that you have enough of a response, an antibody response, to protect you. At least we know based on the studies that when we give you those 2 shots or the 1 shot, that we're going to get to the level that you need to be at.
Dr. Selleck, any other thoughts
Yeah,
on the timing?
As medical literature has evolved, it turns out that people who have asymptomatic or minimally symptomatic COVID-19 develop a much less robust immune response than people who are severely ill. And it turns out that these vaccines actually generate an immune response that is equivalent to that of people who have been severely ill. So just because you've had it doesn't mean that you shouldn't get the vaccine. We were originally saying that you should wait 90 days because we know that you probably have some immunity for 90 days. And when we were very short on vaccine, we wanted to kind of spread it out. But there's, once you're recuperated, you can and should get it. And we recommend that for everyone. And don't be, fearful of, this seems to be, I think we've been locked inside for too long because social discussion anymore is about what kind of side effects did you get with your vaccine. Don't worry about your side effects. I've not seen anybody be severely harmed from vaccine. Some people get achy, but if you look at all of those big trials that I showed you, almost half of people did not have any symptoms when they got the vaccine. So don't be fearful of that, and don't think as I commonly get questions, I didn't have any side effects with my vaccine. Does that mean it didn't take? Forget about Forget about that stuff. Forget about antibody levels. We have no idea what these antibody tests mean. There are companies making lots and lots of money on these antibody tests that are generating bits of data that have no known value at the moment, other than perhaps for doing some epidemiology. So don't run out and get tested. Don't worry about the side effects. Just get vaccinated.
But
I'm
as
reassured to hear you say that lack of a reaction does not mean you didn't get an antibody response, because I had none.
Yeah, no, but I think it's important from the primary care perspective to give people sort of the array of what's going to happen, because people out there were getting shots and then suddenly they have all these other changing into Spider-Man. Then we really have to be very careful. So I think for us, we started. My whole staff was 220 of us. Got it back in December or January, and we went through that whole sort of episode, and we were just kind of getting people to explore, because they become ambassadors for you. That's what I've done. I've created 4,000 ambassadors, and I want them to tell people, yeah, you might have that, but at the end of the day, you're not in the 547,000 people that died category. So and I think it's important. We try to pay. I tell people, you stay 6 feet apart, or you're going to be 6 feet under. So it's just a lot of little things that help on the clinical side, so that people get it.
Yeah, one of the things that you might do is, if you have something really important coming up, like you're presenting at mini medical school, maybe you don't get your second dose the day before, because you may, and we did that in the hospital. One of the nephrologists called me up and said, "How come you're not vaccinating the dialysis nurses?" And I said, of course we're vaccinating the dialysis nurses. We're not doing them all on the same day, because if a bunch of them get side effects, then there's going to be nobody to dialyze your patients. So we broke them into thirds or quarters and spread them out, so that if people were going to be out with side effects, they weren't going to be out all at once. So you can do little things like that, but by and large, there's really nothing that's a showstopper.
So I've heard anecdotally about long COVID, people getting vaccinated after having had long COVID, and it actually improving their symptoms. I don't know how great literature there is on that at this point, but anecdotally, at least, I've heard that at least you don't get worse.
That's right.
But especially the headaches. It's true. Okay. I've seen it with some of the people that follow up.
Yeah, Jen, you said anecdote twice, and that's important. They're anecdotes. I go back to the idea that if you've had COVID, get vaccinated. That's the key. That's the
bottom line.
So there are a number of questions about the ever-changing ages for meeting the criteria for vaccination. We know they vary in states, among states, and they keep on going down. Can anyone comment on the criteria right now in Erie County?
Yeah, so at least in New York State, the criteria is anybody 30 and above. And as of April 6th, it's 16 and above.
Right. So that was a question. So 16 and above as of April 6th, we're almost there. And then that goes to the question that was raised about.
But remember, it's just
Pfizer. There were some questions about the pediatric studies. Dr. Selleck, you mentioned them. And the question was, would you enroll your child in a pediatric study at this time? Do you think it's safe to enroll in a pediatric study?
Absolutely. We have just such a huge amount of data that these vaccines are safe. I think the bigger thing that we're always concerned about in the pediatric studies is how immunogenic they're going to be. So what you're going to see, there's going to be a little bit of dose ranging done with this for the younger kids. So, do they get the same amount of the the live ammo, as it were, that an adult gets, or half or 3/4 or whatever. But I think it's the same reason that we we vaccinate against everything else in kids, and that is kids are often the reservoirs. Now, it turns out with SARS-CoV-2, they're not the main reservoir, but as we get more adults immunized, then kids are going to become the reservoir, and then they will be able to transmit it to other people. So I think that even though kids generally don't have a real bad time, we've seen a number of kids locally and certainly nationwide. There are a few hundred reported with the multi-system inflammatory syndrome that happens with COVID. But I think that absolutely, my youngest is 28, so she doesn't qualify for any of these pediatric trials, but she has been vaccinated.
Mhm.
I was just going to add, because as Dr. Selleck said, because the children have not we haven't had the cases in children the same we have had in other age populations or age ranges. I think it's going to be important to temper people's expectations in terms of what the results of the pediatric trials are going to be. We're probably not going to see these blockbuster results like we did with the adult populations, simply because children don't get the disease as often as the rest of us do. And so it's really going to be more about what's safe, and maybe some transmission data. But it's not going to be preventing. We're not going to have enough cases to compare to for serious disease and hospitalization the way we did with the trials with adult populations.
Yeah, when John Mascola from the NIH talked to us on St. Patrick's Day, in fact, he even said that the endpoints of the pediatric trials are going to be safety and generation of antibody response, that they're not even trying to look at the clinical part because, you wouldn't be able to do the trial in a reasonable time frame to do that. You would need millions of kids in the trial, which is not realistic.
Yeah.
So thank you. There were a number of questions about false positives and false negatives. And in particular, you could rationalize that if you have a negative and then you become positive, maybe it had to do with viral load and how it changed. What about the reverse order? If you've had a positive and then a few days later you have a negative, is that related to variants? Is it related to problems with the test? What can be explanations for that? And can we, since we make decisions based on those outcomes, do you have confidence in the testing?
Well, I think early on it was a problem because of the testing and how we package it, and you didn't know if you had enough virus. By the time it got in 2 weeks later, you had no idea. I think some of the rapid tests, they're not 100%, and so as a result of that, you're going to have to go to the gold standard. And even when you go to the gold standard, the ratios there aren't as accurate. So, I find that if you're testing all the time, of course, you're going to get some variability. And we found that people, once they were positive, we're not going to be repeating a test, again, in a certain amount of time, because it's going to either be positive or falsely negative during that time. I don't know, Dr. Selleck, that's what we were kind of doing out in the field.
Well, it's very important. It's probably, I could clone myself. Maybe I could ask Dr. Surtees to clone me, so I have one of me just to answer all of the testing questions that I get in the average day, because now we're doing so much testing, and the more you do, the more the funny things that you see. So it really comes back to, what is the likelihood that the patient is infected? Now, I understand that there's, 20% of people are asymptomatic, but when you have somebody who comes in to see you and they have fever and cough and they've lost their sense of smell, if that test is positive, that test is positive. And you're going to have to work really, really hard to ever convince me that that's a false positive. I mean, that's the pre-test probability is very high. That's a reliable test. The problem that you have is when you're testing people who are contacts, people who want to go to a ball game or a concert or whatever it is, so they don't have symptoms, then you're at the point where, especially with these rapid antigen tests, which are really underperforming, in my opinion, you're at the point where maybe half of those are false positive. So a lot of times when we get people like this in the hospital and they, it's usually for something other than COVID, and they have these funny test results, we repeat a PCR. And obviously, as Dr. Vasquez said, even though PCR is the gold standard, it is not 100% in either direction. I mean, it's usually reliable if it's positive, but if it's negative, did you get a good specimen? Did you get it from a good place? Did the patient pull away when you're trying to stick this swab into their brain, as everybody thinks it's going into their brain? It's not. We did these on each other in medical school for we just screened our whole class for meningococcal carriage, and it felt like it was a pencil going in my eye. But it's not going into your brain. It's just tickling those nerves. But those are all the things that go into the likelihood that you're going to get a reliable result. And one of the things, it's just an ancillary thing to all of this. The vaccine will not give you a positive test. So don't, if you get a positive test the week after your first dose of vaccine, that is not because of the vaccine.
So let's get to another series of questions, which I think many people are asking now that they've had their second dose. Can we travel? And if you travel, is air travel safer than other forms of travel? What are the safest forms of travel? What about international travel? Is that different from domestic travel? And last but not least, for each of you, would you travel?
Did you ever go to Florida, Roseanne, that time after we talked?
No.
No.
So I'm, so I've been, my whole family's been vaccinated. So we're going to be traveling on Friday, but we're going to follow the logistics. We're going to Jamaica. We'll wear a mask on the way down. We'll do everything else. And we're going to be pretty much together in those activities. So we'll but I think we have to get back to a little normal. I'm not saying you're going to go out there and just go crazy.
Yeah.
So, but I just think we have to, adjust. I mean, when we go out to restaurants now, everyone's been vaccinated. It's a group of people that you normally hang out with. And so, we don't wear masks when we're eating. And that's been our kind of protocol. We started with one thing and just got a little bit more and more as we felt more comfortable. But yeah, I don't know, Dr. Selleck, so I'm doing the wrong things, but I want to, at one point you want to live,
So I'm not going to call you out in public. I mean, that's not going to happen.
I'm being honest, because you'll see me.
I think this is another very common question. So there's, again, hate using the lingo, but there's another place that applies. There's a lot of moving parts with this. I think if you can travel in your own car and you're very careful when you go to the, rest areas and stop for gas and all that kind of thing, that is probably safe, presuming that you're going to spend time with other people who are either vaccinated or have bubbled so that they're not, that they're not exposed. Because again, we don't have 100% assuredness that we would not, with these vaccines, that we would not still get infected. You don't know if you're in the couple percent of people who don't respond to the vaccine. So you've got to, it's a risk benefit. I think the problem with air travel, and you can go online and you can look, there was about 30 or 35 years ago, there was an outbreak of drug-resistant tuberculosis on a 747, and your risk of getting infected depended on how close you were to the guy who had it. He was the second row from the back in a 747, so he's in the real cheap seats. So from that point on, all of these large aircraft now have HEPA filters, so all of the air circulates through, but of course, that doesn't help when the lout sitting next to you has his mask off and is coughing on you. So you can have all the HEPA filters you want for the air movement. The other big part of air travel is you've got airports, you've got waiting online, you've got waiting in the, in the runway going down to the plane, you've got waiting for your luggage. All of these things that are going to put you in contact with a lot of other people. Plus, when you're traveling, especially if you're going on vacation, you're probably going to wind up in restaurants and you're going to go to, other, activities and entertainment and such. So you really have to put it all together and say, what am I willing to do? If I'm a kidney transplant patient, I'm not. If I'm a healthy 20-year-old and I'm thinking, well, what's the downside here, except if you're going to bring it home to your family member who's a kidney transplant patient. So you really have to think it through. There's not one simple answer for all of this.
Right. So if you are at risk, be cautious because you can't control people around you. If you are in a situation where you can control your travel, a car, or you could avoid restaurants, be sure to wear your mask and possibly wear your goggles if you're going to be in crowds. Because again, right now, I think in Erie County, 30% of our population is immunized, 70% is not. So even though you yourself may be immunized, you can't count on other people being immunized yet. Although that's going up, that rate is going up. We are going to be wrapping up soon, and we will get to some questions that were in the chat. There was one very specific one about whether after vaccination, are lymph nodes enlarged after vaccination? And if so, would they affect the results of a study like a mammogram if that was done soon after vaccination?
I've got males mostly that wherever they got vaccinated, they had lymph nodes. Lasted for about 10 to 14 days, but no females. I didn't see it in females. Yeah. But we did get. I got about maybe 10 or 15 people that said, after the shot, I'm feeling this thing here. It hurts, and so that's. I don't know if Dr. Zellick, if you've seen it with some of the people, because you have more people vaccinated, but we did see it with some of the patients that we vaccinated.
Yeah, it's actually there was just I didn't read the whole paper. I just saw the press release on it. It was on one of my listservs about this is a real good thing if you get enlarged lymph nodes, especially in your axilla, because that means that you've got lymphocytes that are doing what they're supposed to be doing, which is good. I have gotten this question a number of times about the mammography. There is no official guidance from anywhere, but what I did is I went and I looked at about maybe 10 or so of the major cancer centers around the United States, and guess what the answer was? Pretty much whatever you want it to be. There's some that say wait, some that say just let us know. We know how to read the mammogram if you've had the vaccine. Some of them say wait 6 weeks. Some of them say wait 6 months. It's absolutely all over the place and absolutely useless, from the point of view of being able to advise anyone. I think one of the things, just as a sidelight, and this is really right up Dr. Vasquez's alley, in the primary care setting, we've seen so many people during the pandemic who have turned off their health maintenance. They've stopped going for medical appointments. They've not had their mammograms. They've not had blood work, et cetera, et cetera. So I think that, if you finally have this set up, I think I would be inclined to go and just make sure that you tell them that you've had your vaccine so that someone can take that into account when they read the films.
Thank you. I think that's an excellent point to end on. We can't forget the importance of our health maintenance and our primary care. And so it's time to get back to your physicians. It's time to get to your dentist and make sure that you are staying as healthy as you can. If you're not taking care of yourself, that's another risk factor for not having a good outcome with COVID. So please do all that. We still have some more questions in the chat. We'll get to some of them, as I said, and post them online. More vaccine is coming, so although there's no mandate to get vaccinated, get vaccinated. Take it as an instruction from all of us in this session tonight. So thank you all for joining. We hope that you learned something new. Thank you very much to our speakers. Wonderful. Thank you to Jen Britton, who helps us from the Alumni Association and makes this feasible. And again, thank you to UBMD for supporting this session. And I think we're going to have UBMD sing us out in a few seconds. Thank you and good night.
Wellness Wednesday is brought to you by UBMD Physicians Group.
Multiple sclerosis, or MS, is a progressive and inflammatory disorder that disrupts the connections between the brain, spinal cord, and the body. We don't fully understand MS, but we know that genetic and environmental factors play a role. Symptoms include vision problems, dizziness, bladder and sexual problems, weakness, and memory and mood disturbance. We cannot predict the severity of symptoms. There is no cure for MS yet, but research is ongoing. Right now, we focus on managing relapses and symptoms. There is an emphasis on wellness, good diet, regular exercise, good sleep, and social support. MS is a complex disease and needs comprehensive treatment. It is unpredictable, and a medical team that you can trust is important. Together, we can beat MS.
Wellness Wednesday has been brought to you by UBMD Physicians.
Just as a reminder, this session was recorded. It will be posted on YouTube. So you can review it at your leisure or share it.
Thank you.
Thank you. Jen, any final words? You're muted.
I'm sorry. I just said thank you very much for attending tonight, and just be on the lookout for our upcoming invitation for April, May, and June. Have a good night, everyone.
Good night.
Good night.